I'm Jim Jarvis. I'm a pediatric rheumatologist from Buffalo. So I had some questions about the rheumatic fever study. First, the place where you're seeing the dot is the Oglala Sioux and the Cheyenne River Sioux in South Dakota, very high prevalence rates of rheumatic disease, which you might guess since many of our people are living in 3-bedroom homes with 25 people living in there. So the poverty and the crowding are probably not bad genes, are probably what are making our native people sick. I had a question about the choice of the exon array. Matt Morano did this beautiful paper in Science way back in 2013 where he shows that for almost every complex trait, pick it, atherosclerosis, juvenile arthritis, preterm labor, type 2 diabetes, 90% of the genetic risk is in the non-coding genome. So why, for a complex trait, search the coding genome when that's only going to pick up, at best, 10% of your genetic risk?
We're hoping to find something functional. And I think a lot of what's in the introns are in the traditional GWAS studies. Really, there's a lot of thought that they're in linkage disequilibrium with something else.
There's enhancers in introns. There's non-coding RNAs that come off introns. There's CTCF binding sites in introns. There's all kinds of great stuff in introns. Even better is the intergenic regions, where there's also lots of insulators, enhancers. Gee whiz, we're learning that the genome is a busy place.
Yeah, I completely agree with you. In some of our Mendelian studies, especially holoprosencephaly, we are looking at insulators and enhancers and doing a lot of work in that field, and it's tough. We're not coming up with a lot of answers. And I think the effect sizes of just regular GWAS studies are so modest. I think part of it we were hoping to get lucky, and maybe we might have to go to a different chip. There's something I didn't say, is that we actually had some extra exome chips laying around, and there was a lot of heated debate in our lab, but I thought I'd leave that out, but since you asked.
Yeah.
Can I make a point about that too?
Yeah.
So for example, one of the studies that we have going on, as I mentioned in the slides, is on preterm birth, another certainly complex disease. And we do genomes on those folks, and we are looking at both the exonic and non-exonic, all the exciting things you talked about. I think although there's certainly evidence that a lot of the answers are in the non-exonic regions, one of the big challenges, as Paul said, is how do we interpret that and how do we functionally design studies? So I think it does, the old analogy we always use is that geneticists are very good at looking under the spotlight for what they can look at, and that's because that's usually what we can interpret and design biological assays around. Not necessarily that that's the answer there, but that's something that we can start to understand in the nearer term.
Yeah, why are you looking here for your car keys when you lost them over on Lombard Street?
Yeah,
well, the light's better here. But
exactly.
Yeah, I think we're going to learn, we have lots of fun stuff looking at the non-coding genome. It's a little bit harder to look at, but it's, attack sequencing and things like that, or at least give us a nice way to do this without a lot of cells. So, we'll get there.
I think the other point is that maybe all our DNA hypotheses are totally wrong. We should be focusing on the microbiome and epigenetics, and then all So there's
Well, Ed did those beautiful studies a few years ago, showing how even with single Mendelian traits, things get really wacko.
Yeah.
It's not as simple as we thought. So obviously, as I say, the genome's a very busy and interesting place.
Yep. Keeps us in business.
Luca Brunelli, neonatologist from University of Utah, but originally from Italy, as my accent might tell. I just have a question or a comment for everybody. So I noticed in the first presentation, Dr. Solomon, the sort of a nice history of genetics and how things developed over the years. I think that in thinking about precision medicine and some of the things that need to be put in place to really launch the field forward even more, I noticed that sometimes we forget the all great influence that recombinant DNA technology has had on the field, and the fact its invention at Stanford in the 70s really was the first thing that allowed us to link phenotype to genotype and have in our hands, through DNA cloning, regions of the genome that we could functionally analyze, including, as the first gentleman was mentioning, that these non-coding regions are so interesting but so difficult to analyze. So I guess if I can be a little provocative here, I would say, it seems to me that we're putting so much effort on building computer networks and doing a lot of sequencing, where, for example, we're still missing a technology to clone specific regions from genomic DNA of individual patients. And it seems to me that this step is very important to allow us to move forward and interpret in individual patients the significance of variation that we can find both in coding and non-coding regions. I think that until we have that technology, a lot of what we do has limitations because we don't have a quick way of moving from structural resolution of the genome to a functional analysis in individual patients, which is what we're trying to get with precision medicine. Any comments would be?
I'll make one comment, and then I'm sure the other panelists have smarter things to say than I. I think there's lots of views on this. Someone that Paul and I both know think that all this genomics, we should not be doing it. There should be one lab dedicated to each gene or maybe each portion of the genome, and that lab should function on really understanding that and not get messed around in all the big data questions. I'll say that very briefly, but perhaps some of the other panelists could comment as well.
Yeah, I think that's an important point, and I think that that's the focus of a lot of the research that's being done with induced pluripotential stem cells. That, if you take a skin biopsy from an individual patient with a genetic condition, and then you make stem cells from that skin biopsy, then you have the capability of studying the biology of that particular patient, and that's a practical and feasible way of doing that. So I think that technology is already available.
That technology is certainly available. I guess what I'm talking about is really the ability to, let's say I'm a patient with some disease and I get sequenced and some abnormalities are found in this region on chromosome 8. But those are now, but those variants, unknown significance, however we want to call them, have not been linked to any specific disease. So if I want to functionally study a large region of my genome, right now we don't have a technology that enables us to come in and grab, retrieve that segment from my genome, clone it, and study it. I mean, that is currently unavailable, and that I see as a critical sort of linking, a critical barrier to advancing precision medicine even more.
I had another
Please, can you introduce
completely
yourself, too?
opposite perspective. Golder Wilson from Texas Tech. This is the third excellent genomic symposium that has viewed the genomic information as an additional burden on the pediatrician rather than placing the pediatrician, who's very informed about genetics, at the center of the medical home and conveying this information to the family the way they do for almost all pediatric disorders. And I wondered what the panel thought. Are you more on the side of Mike Bamshad, as he expressed 3 years ago, that this should be coming from the commercial company or the scientists through a genetic counselor to the patient? Or do you feel the pediatrician might have a role, and that to be mature, as Dr. Bianchi said, for the prenatal genetics, a mature genomics will be when the informed pediatrician is able to comprehend and relay this information?
Yeah, I think that's certainly a tension. But the general pediatrician has an awful lot going on in their offices today, and we have to figure out how to take a very complex topic and turn it into an image or some way of reducing the data so it's understandable. And you can see a practicing pediatrician, a practicing geneticist can figure out how to use this information. So I think the challenge is really in terms of how we represent the data, so that it's more understandable. But along those lines, there was a study that came out of Children's National, Marshall Summers group there, looking at newborn screening. And what they found was that in DC and Maryland, where the laws and regulations allow the newborn screening lab to talk to the subspecialist and the pediatrician of record at the same time, there was a decrease. I can't remember, it was like 3 or 4 days compared to Virginia, where the regulations only allow contact with the primary care physician. And then there was a huge delay in them making contact with the specialists, the genetic specialists. So it's just one example where I think we all understand the plight of the general pediatrician with so much information now, so that, again, you know, I'm not a practicing geneticist anymore, and to me, you know, I see this, I understand the terminology, but I don't really see it in my mind. And so we need to make it so somebody like me can make sense out of it.
I totally agree with you. I deal more with the obstetricians, although I'm a pediatrician. My expertise is in prenatal genomics. But the obstetricians and the pediatricians share the same problem, in that they are getting the test results, and yet they don't have the educational background to give the appropriate counseling. And so I put the burden on, you know, education, either undergraduate or postgraduate medical education, but it's not keeping pace with the technology. And we're not talking about research anymore. That was the whole point of my talk. We're talking about this, anybody can get this test. So who's going to counsel them? There aren't enough genetic counselors in the country. There aren't enough medical geneticists. So the technology and the pressures pushing the technology forward are greatly outpacing our ability to provide clinical care. In the ideal world, it should be the primary care providers, but who's going to educate them? And that's the dilemma. So we're, at least in the genetics field, dealing with new models for education. It's going to have to be online. Nobody has the time to take a day off or a week off to get updated in these you know, very important principles of genetics and genomics.
Hi, Neil Romberg from CHOP. Really enjoyed all of your talks, appreciated the breadth and the depth of the seminar. I have a question, actually I have 2 questions for you, Dr. Bianchi, about cell-free DNA and sort of the operating characteristics. The first is, what are the challenges of dealing with this procedure in a woman with multiple fetuses, sort of triplets, quadruplets, octuplets? Is it possible? Is it just like Sudoku, a lot of math?
Okay, so multiple gestations, I didn't have time to go into them. The test is not recommended for more than twins. And the limitation with twins, although the test is offered with twins and there is some information on performance data in twins, it's kind of interesting that the fetal fraction, meaning the amount of circulating DNA per fetus, is not double in twins. So fetal fraction is the amount of fetal DNA, fetal placental DNA over the maternal DNA. And you would think it would double, but it doesn't double. So you have a relative lack of fetal fraction DNA per fetus. So the way I counsel people is if you have twins, you can have the test, you have a slightly increased chance of having a false negative result. I don't worry about false positives. When you get a positive result, you don't know, is it one twin or both twins if they're identical, but it's a screen. So if it's a positive result, you go on to the next step and you would have double invasive procedures. The only thing I worry about with twins is a false negative, and the test is not recommended for higher order multiples.
Thank you. And my other question is about the resolution of picking up deletions in the method. I think you had said that you could that the method could pick up deletions of 7 megabases. Is that right?
That's the current.
Okay.
It's not. There's a non-branded talk, but Sequinome is the only one offering that test.
Okay.
So they're offering it at the 7 megabase level.
Okay. So I order chromosomal microarrays often on my patients, and the resolution is much better. Is that a difference in technology or a difference in what Illumina is willing to say they can detect?
I can't speak for Illumina or Sequenome, but clearly if you're ordering a chromosome microarray on an individual, you've got pure DNA there, so you don't have the issue of the maternal background. And you can increase your resolution if you increase the depth of sequencing. So that's I can spend a whole hour just talking about non-invasive prenatal testing of microdeletions, but it's mainly related to the fetal fraction, the depth of sequencing, and the region that you're trying to identify, which is influenced by the underlying base content and other things. So most people, I mean, we actually had a publication in the American Journal of Human Genetics where we detected a 300 kilobase deletion, but our sequencing depth was something like 125 million tags, something like that, which is ridiculous. It's very expensive to do that.
Thank you.
Hi, thank you to all the speakers for very excellent talks. My name is Bina Kamath-Rain. I'm a neonatologist, and I do research in the amniotic fluid transcriptome and fetal maturity. But I actually wanted to ask the question as a parent. I'm a parent of a child with an undiagnosed disease, and we've had a bunch of genetic testing done. And as a parent, it's actually been a struggle to figure out who owns that data, and if I was tested along with my husband and my child, feeling like the institution where I'm located wouldn't let me have ownership of that data to get it to other networks or other institutions that might have the ability to look at it differently. And I was wondering what recommendations you have for pediatricians or parents that want to advocate for their child to get the data where it needs to go so that they can get answers.
I'll start, but I think that's a fantastic and very important question. I'll make a couple comments, but I guess some of the other panelists might also want to respond. So a lot of these questions are, I don't want to say unfortunately, but are often relegated to what the original consent and the IRB document said, right? And there's a latitude depending on the institution and the IRB and the investigator certainly about what can be done. There is often within certain protocols flexibility to allow some of this, and I would also say that there are endeavors at our institute and many other institutes that advocate for more some of these data sharing and for basically, this is going to sound a little glib, but crowdsourcing of experts within specific conditions. So for perhaps an expert that really thinks deeply about the bioinformatics of the condition that affects your child, and there are a lot of endeavors that are working to do this. Having said that, this is a really tough problem because, of course, folks want to do the very best for patients and families, but there are all sorts of privacy and questions that, again, just like you've heard in these talks, we're very early at thinking about these and amassing the evidence and the data and the best practices along those lines. I'd be happy to talk to you more without kind of revealing any personal information, though, afterwards, if that would be helpful about specifics, okay?
Yeah.
Yeah, I think it's, first of all, your institution needs to be more open. I'll just flat out say it. There's difference of opinion about research genetic data within the federal government. Whereas part of the federal government, HHS, says that basically institutions are stewards of the data, but don't own the data. Whereas the courts have said that the institutions own the data. So judiciary and the executive branches disagree. The other thing, though, is I think it's the ACA has required by some point in time, which is relatively soon, that every patient have access to their data through a patient portal. So you should have full access through a patient portal, and that is mandated by the ACA. So if you don't have that, then you need to find somebody who understands that law much better than I do. But I think it's very soon, or may have passed, that basically requires patients to have access to their data.
will get them data through the physician.
Yep. If there was certain funding, such as NIH funding through the Freedom of Information Act, there are also mechanisms that way to try to get access to the data, depending if it was research or clinical. But
Yeah, I think it matters whether you enter through a research portal initially or a clinical portal. So I've dealt with it both ways. Some of the unusual cases that I was describing in my talk came in through a clinical portal, and their consent didn't say anything at all about detecting trisomy 7 or trisomy 20. So we had to consent them secondarily through a research protocol to be able to look more deeply at their genomes and then return results to them. But I know that some of the, I mean, I had my genome sequenced and I was given a hard drive with it. So there are ways you can do it if you're willing to pay. I just did it out of curiosity, but so we can talk offline about that.
I think that's an important topic. I will say there's a lot of movements in the genomic community to return or to have patients control their own data. Even research, not just clinical or CLIA approved, there's groups like Genetic Alliance and groups that are really trying to concentrate this. Because what happens a lot, and this happened in our institution, where a principal investigator retired, and the program stopped, and where does all this data go? And I think that some of these organizations, like just pick an organization, let's say the Turner Syndrome of the United States. This organization would maybe control some of this data, where the parents would have control of that data, and the consenting process would be a spectrum that would go on, and you would consent, and they would somehow control all this research data. Obviously, a lot of investigators are going to be adverse to this because, there's a sense of control when you have biospecimens or stuff in your research data on your computer hard drive. But anyways, yes, there is a lot of movement in what you're talking about in parents and patients getting together and controlling their own data. And that's my prediction. In the next couple of years, we're going to see the paradigm of research change a lot because of that.
Great question.
Sorry, I'm a little short. Hi, I'm Talia Schwartz. I work at Boston Children's Hospital. My question is sort of about legality as well in terms of GINA and the protection of genetic information by insurance companies. And as we grow and we do more whole exome, whole genome sequencing and tend to find incidental findings for children and adults, how the legal system will also incorporate the transition of medicine and expand its legal protections by insurance companies.
Yeah, that's also an important question. I like to use as an analogy with GINA that civil rights legislation was passed when I was quite young and is still litigated. Until we really litigate through GINA, we're not going to know how much of a protection it is. And there are parts of in the insurance business that are not impacted at all by GINA, for example, life insurance. But I think GINA was important. I don't want to denigrate GINA. We're safer because of GINA. But until we know the extent to which the courts are going to allow GINA to be a part of our daily lives, we don't really know its full spectrum.
Just if there are non-geneticists here, it's the Genetic Information Non-Discrimination Act. That's what GINA is.
I know one of the investigators at NHGRI, a bioethicist, has filed a Freedom of Information Act to get all the current GINA records, and one can view some kind of cursory data about that, but he's looking very specifically into it, but completely in agreement with Dr. McCabe. It hasn't been challenged very much yet, and there's not a lot of knowledge about its applications or its tests yet. I think we're done. Thank you so much. I want to, before you guys leave, I think what I would like to maybe just ask very quickly each of the panelists to just mention briefly one thing. It doesn't have to be the thing, but that they're most excited about, with the current genomic technology and with the field. And so to give them a little time to think about that because they're not prepared, I think the thing that I'd like to mention that kind of excites me about this field is what Dr. McCabe talked a lot about with groups like Stephen Kingsmore, formerly of Kansas City, now of San Diego, is providing diagnoses for children with conditions much more quickly, right at the bedside, so they're not going through these long diagnostic odyssey processes.
And Stephen Kingsmore was just entered into the Guinness Book of World Records for sequencing the fastest genome. So what I'm most excited about is really the opportunity to treat prenatally. So I do foresee. I don't know if it'll be the newborn screen in the 3rd trimester, but I'm hoping that a lot of this genetic information and genomic information that we're obtaining prenatally will be used to initiate therapies that we can offer to pregnant women when they find that there is a diagnosis in their fetus, and that will result in healthier babies being born.
Yeah, what I'm most excited about, I did mention it in one slide, and that is the integration of the omics. I think we have an opportunity to bring very different streams of data together, but again, reminding you that's the electronic health record, the images that are now digital, as well as the genomic and metabolomic data that are available to us. But I think when we have the ability to combine those, it will be so much more powerful than any single stream of data that we've experienced in the past.
I think what I'm most excited about was an underlying theme of everyone's talk, except for mine actually, was reduced penetrance. And I think trying to figure that out, it's phenomenal how unpenetrant all these genetic diseases and genes are. It's amazing, and I'm real excited about all these new genomic databases and a lot of the work that's being done in the lab to figure this out. I think as we figure, looking at knockout genes and in big data sets like the exact and decode, I think a lot of these answers are going to be solved.
Thanks so much for your patience and for coming.